Hey there, welcome back for more biotechnology. So last time we had a pretty lengthy discussion on the different sources of DNA, how there are physical DNA differences and regulatory differences depending on whether you're getting your DNA from bacteria, eukaryotes, or viruses. So now we're going to talk about something a little bit more fun, and this should be a fairly short video, but we're just going to have an introductory discussion on isolating and manipulating DNA. So at the end of the last video, we had this discussion here on genetic engineering, and really it's the same sort of discussion that we had previously on using recombinant DNA. But now that we have a little bit of a better foundation about what DNA is all about, the central dogma and things like that, we can talk about this in a little bit more detail. Like I said, this should be a fairly short video. We're not quite ready to have more detailed discussions on this, but we can at least go ahead and talk about it a little bit more. So we talked about gene therapy, the idea of correcting a defective gene. So this leads into a discussion on a method of changing the sequence of DNA, something called site-directed mutagenesis. So let's talk about that. So when you're using recombinant DNA, in many cases, the gene that you're trying to introduce into a new organism is going to be what we call the wild-type version of that protein or that gene, meaning that the protein that is made from the gene you have transformed bacteria with or transfected eukaryotic cells with, if it's wild type, that's going to be the same protein amino acid sequence-wise that is found in most members of that particular species. So if you transform bacteria with the wild type version of insulin, the insulin that the bacteria make is the exact same insulin that almost every healthy human makes all the time. So wild type genes and wild type proteins are different from what we call mutant genes and mutant proteins. Now, the word mutant and mutation has been kind of wildly overinflated in popular culture. So when people think of mutants, they're thinking of things like the Teenage Mutant Ninja Turtles. You're thinking of some slimy organism that has all these tentacles and things like that. So that's not what mutation means. Literally, a mutation just refers to a change in the sequence of DNA. That's all it means. But a change in the sequence of DNA can have some very drastic consequences because since the DNA sequence provides the instructions on how to make proteins, if you change the DNA sequence, you're probably going to change the protein sequence too. And if you change the protein sequence, you're going to change a lot of things about that protein and therefore a lot of physical characteristics about the organism potentially. So when the DNA sequence is altered and the protein's amino acid sequence therefore changes, the protein is what we call a mutant protein. So we've already covered the idea that in sickle cell disease, these people that have this disease make a mutant form of the beta-globin protein because we have changed the DNA sequence and therefore changed one of the amino acids. So if we wanted to use this technique called site-directed mutagenesis, what we can do is we can actually change a nucleotide in the source of DNA in order to mutate the DNA from wild type into some mutant that we want, or if we're talking about gene therapy, we can revert the mutant DNA back to the wild type version of the DNA by changing the mutant nucleotide back to what it is supposed to be. So site-directed mutagenesis is useful for making what are called point mutations. A point mutation is a change in a single nucleotide. Now, this doesn't really have anything to do with some more widespread mutations like deleting whole sections of DNA or adding whole sections of DNA, flipping DNA sequences upside down. So this is from just changing one nucleotide at a time. So like I said, because the DNA provides the instructions codon by codon for how to put amino acids into a protein, a single nucleotide change may change the amino acid used in the protein according to the genetic code that we talked about in chapters 1 and chapter 2. So look at this chart right here. So if you look at the DNA level, let's say we're dealing with the codon TTC. Well, in mRNA, the mRNA that is transcribed off of that DNA, that would be AAG. And if you look at the genetic code, AAG will code for the amino acid lysine. So let's make our first mutation here. Let's say that we change that cytosine in the DNA into a thymine. Well, now the mRNA is not going to give you the codon AAG. It's going to give you the codon AAA. However, that is not going to change the amino acid that is used, because the codon for lysine can be either AAG or AAA or several other codons as well. There are redundancies there. So if this is the type of mutation that you make in which you change the DNA and the message RNA but not the amino acid, that is called a silent mutation. A silent mutation changes the DNA sequence but not the amino acid sequence. So there should be no defect in the protein itself, and therefore there should be zero impact on the health and function of the organism, because it's the protein that's doing all the heavy lifting, not the DNA. The DNA is just the instructions on how to make the protein. Look at this next one. So this time, if our original codon is TTC, let's change that T to an A. So now it's ATC. Well, the messenger RNA transcript is then going to be UAG, which is a stop codon. This is a big deal. This is what we call a nonsense mutation. What this does is it takes that lysine that would have been inserted into the protein, and we insert a premature stop codon. This is going to cause the protein to be way, way, way too short. We should have inserted the amino acid lysine plus any other amino acids that would come after that, but because we prematurely put in a stop signal, the protein is just going to stop right there, and it's going to end up being too short. It's probably going to misfold, and the cell is probably going to dispose of it. A very good example of this is the disease cystic fibrosis, in which there is a mutation into the CFTR gene that causes a premature stop codon, and this chloride transporting channel, which should be aiding in the mucus formation in the lungs and in other places in the body, it does not get made properly, and people suffer for it. The last and most common type of point mutation is called a missense mutation. A missense mutation is another point mutation that actually does change the identity of the amino acid. In this case, we can have two types of point mutations, or two types of missense mutations. A conservative mutation will change the amino acid, but we're changing the amino acid into one that falls into the same class. So both lysine, what would have been the amino acid, and arginine, those are both basic amino acids. They are very similar in their chemistry, and even though the amino acid is now different, the change isn't that drastic, and maybe the organism will be okay. But a non-conservative mutation not only changes the amino acid, but it's an amino acid of a completely different chemical class. Threonine is a polar amino acid and has no basic function. It has no ability to interact with acids. So that actually may have dire consequences for the organism that has to deal with it. Since we mentioned sickle cell disease, that would be an example of a non-conservative missense mutation, because a aspartic acid residue, an acid amino acid, is changed into a valine, which is a hydrophobic amino acid. So that is a big deal, and the protein ends up not folding correctly as a consequence. So site-directed mutagenesis allows us to, once we have our DNA isolated from whatever organism it came from, we can alter its sequence one nucleotide at a time. But in order to do this, we need to use a technique called polymerase chain reaction, which we mentioned the guy who invented PCR. His name was Kary Mullis, but we haven't really discussed how that works exactly yet. So we're going to table this discussion for now, but once we do start talking about PCR, we will be sure to revisit the concept of site-directed mutagenesis. But regardless, hopefully you should be able to appreciate and see how useful site-directed mutagenesis would be for gene therapies that would treat diseases that are caused by point mutations like we have discussed. So that's going to do it for this short-ish video on site-directed mutagenesis and manipulation of DNA. So join us for the last video in this chapter, and we are going to start talking about gel electrophoresis for separating and studying DNA molecules. A very exciting discussion and something that we will actually be doing in lab. So I hope you'll join us next time.